Good evening, ladies and gentlemen. May you all please rise for the academic procession. Thank you. May all be seated. Thank you.
Colleagues, good evening, friends, good evening. My name is Bertie Fielding. I'm the Dean of the Faculty of Science. A heartfelt welcome to everybody in this venue today. A special welcome to our DVC Teaching and Learning Professor Ramjankanath, DVC Research and Innovation Professor Moyo, School Director of Data Science, Professor Kanshu. Colleagues, I cannot greet all our dignitaries by name because I look across the audience and there are many, many, many of you. So please, all of you feel welcome, all of you feel appreciated. We thank you for being here. Tonight is really a celebration of that man on the corner there in the red gown. But I would be amiss if I do not give a special welcome to Thulani's grandmother, Anna, his mother, and his father. Thank you very much for being here. We welcome you, and I think I can speak on everybody's behalf. You've done a great job in raising not only a great researcher, but an amazing person and man. Thank you from our side and congratulations. But we all know, there's a saying that says behind every successful man, there's a great woman. So Thulani's wife, congratulations. Thank you for being here. Thank you for keeping him in tow and for just making sure that he does what he needs to do. And then his two daughters. Welcome to you as well. You can be very proud of your dad, because we certainly are. Colleagues, we have 150 RSVPs for tonight. It's one of the biggest groups we've ever had together in our faculty. And it says something. It says something about the man that's over there. Professor Alf Botha will do the introduction. So I've been told I can only keep it to 5 minutes, so I can't say too many things. But I think Thulani is the epitome of what a researcher and an academic should be. Only if you have and you lead a balanced life can you look like that man who smiles all the time. I've never met anybody in my entire life that smiles that much. When I look at him, my cheeks go lame. So, Thulani, you an inspiration to the students that are here today. You're an inspiration to your colleagues, and you're even an inspiration to the leadership of Stellenbosch. So once again, from my side and from the faculty side and the university side, congratulations. We know that you're excited to do this talk. I don't know why you're nervous, by the way, but we're looking forward to this. Congratulations. Alf.
Thank you, ladies and gentlemen. Good evening, distinguished guests, colleagues, students, and members of our community. It is an immense honor for me today to introduce tonight's speaker, Professor Thulani Peter Makhalanyane. We celebrate, for us to celebrate his inaugural lecture here. It's a great honor for me. This occasion marks a significant milestone in his academic career. Let's take a moment to reflect on this, on his contributions, what he has done so far, and what is going to happen in the next few years. Professor Makhalyane's expertise lies in the intersection of microbial ecology and data science, making his contribution uniquely multidisciplinary. He is an internationally renowned expert in studying microbial communities in extreme environments. Environments like the frozen deserts of Antarctica, the scorching hot Namib Desert, the unforgiving Southern Ocean, and the hidden biospheres beneath the Earth's surface. The environments, in these environments, microbes persist, and they survive there. They survive and they enrich our environment there. Okay. So, and by studying this, Professor Makhalanyane basically gave us the questions or the answers to questions that actually we are struggling for many, many decades in the past with his research. But before I delved into the themes of this lecture, it is important to appreciate the journey that has brought Professor Makhalanyane here. He obtained his PhD from the University of the Western Cape, where he was recognized as a very hard-working scientist and always enthusiastic about his science. That is so. We all know him like that. His postdoctoral journey took him basically all over the world, towards all, how can I say, the most prestigious institutions over the world. And what he did was, he didn't just go there for holiday. No, what he did, he studied. He studied and he gained experience in the fields of genomics and metagenomics. Techniques that allow scientists to study entire microbial communities rather than isolated species. It's very important what I'm saying now. It's a new science. These tools are crucial to understanding the complexity of microbial ecosystems in their broader impact. That's extremely important, guys. We can't understand nature with these things without this. Beyond primary focus in microbiology, what makes Professor Makhalyane's work particularly pioneering, and that's why we're very proud of this, is the joint appointment of Stellenbosch University School of Data Science and Computational Thinking. It's very important. This dual affiliation underscores his belief in his belief in the power of interdisciplinary collaboration. I can't even pronounce this. And his integration of different fields. This is also important. And so by doing this, you can actually learn more about nature. Different fields of research combining, and then you understand better. He has perfected it, guys, really. So this fusion of biology and data science is essential to answering some of the most pressing questions of our time. Questions about biodiversity. We all know about biodiversity on everybody's lips. Climate change, yes, is involved there, and ecosystem resilience. Moreover, Thulani's research highlights the critical connection between environmental microbes and human health. Yes, us. Just as the human microbiome, the community of microorganisms in and on our bodies, affects our well-being. Environmental microbiomes impact on ecosystem health. which is very important, and we are part of that ecosystem whether we like it or not. So, by drawing parallels between these two areas, Professor Makhalyanye's work prompts us to reflect on how deeply interconnected human life is with the natural world. We are part of it, whether we like it or not. In addition to his research, Prof. Makhalanyane has played an influential role as an educator and mentor, shaping the next generation of scientists. His students consistently speak of his passion for discovery, his commitment to academic excellence, we ourselves can vouch for that, and his dedication to fostering an inclusive and diverse scientific community. In less than a year since his arrival at Stellenbosch, listen to this, his research group grown from 0 to 18, less than a year, postgraduate students and researchers. How's that? From 0, eh? Professor Makhalanye's work, it is nice. Professor Makhalyanye's work, however, extends far beyond the confines of the laboratory and classroom. His thought leadership is being sought by national and international organizations. He's always on the plane. And he has contributed to important global conversations on the environment, science policy, and the role of data in driving innovation. Recently, he successfully organized the 19th International Symposium on Microbial Ecology. It was the first of these conferences on the African continent, and delegates from around the globe attended this prestigious event. Also, I have to say this. Since his arrival at Stellenbosch, less than a year ago, Professor Makhalanyane authored 12 articles, okay, in high-impact scientific journals. 12. Where did my 4 come from? Including in the journal Nature. And they've got Stellenbosch's address on it. I have to say that. It is now my great pleasure to welcome Professor Thulani Peter Makhalanyane to the stage for his inaugural lecture, named Unveiling the Hidden World Within Us, What Environmental Microbiomes Can Teach Us About Ourselves. Please join me to give him a warm welcome.
Thanks so much, Alf, for the introduction. I was wondering, we were chatting earlier today, I wondered who was more nervous about today, Alf or me? We'll soon find out. Yeah. So, as Alf said, it's almost a year since I got here. I started in November, and I remember when I made the announcement that I'll be moving to Stellenbosch University. Everyone, I got tons of congratulations, including congratulations spelled with a D. And then there was one person called Engineer of Sense who said I should tell us after 2 years if you're still there. Well, Mr. Engineer of Sense, I'm still here. It's only a year. I'll let you know how it goes. But hopefully next year I'll still be here. We'll see how it goes. So, of course, inaugural lectures are about celebrating one's academic achievement. So I've been quite fortunate in my career to work with brilliant people and put out lots of publications. But I thought, at the start of such an occasion, which is of course about celebrating academic excellence, it's important to know that, of course, in academia, there's tons of failure, right? This is just a screenshot from my Nature publication, and you can see they're highlighted in red. Of all the publications that are published eventually, there's 39 declines, right? And there's a lot more of those declines out there. So as we celebrate this, it's really good to put in perspective that, of course, academia is full of more failure than sometimes success. So this is the outline of my talk today. I'll talk about the Earth as a patient in need of healing. I'll talk about the Microbiome and One Health, microbiomes and sustainable development goals. I'll talk about the advance of genomic sequencing, and then I'll discuss extreme environments as a good model system, and touching on the work we've done in Antarctica, in the oceans, and also on the African continent. And then I'll put a summary together of some of the lessons that I've learned from studying environmental microbiomes, and then talk a little bit about what I hope will be the next 2 decades. And if engineer of science, or whatever he's called, is here, I hope those 2 decades are here. So, all right, so maybe a brief overview of my journey to exploring the hidden world around us. This is me as a child. As you can see, I was generously proportioned. My grandmother took very good care of me, and I got lots of pap, and it shows. I had a really happy childhood thanks to my parents, who kept me nourished in many ways, intellectually, and spurred my development. And this is me at my first graduation. As you can see, I was really happy about it. Yeah, you know, I've since learned to smile, as you can see, and I think it makes for a much better look. So, yeah. So my parents had thought, like, you know, many black parents, that if you are gonna do anything that's worthwhile and, you know, comparable to success, you've got to be a medical doctor. So sadly for them, that's not the path I took. But I'll tell them I am sort of a healer, but I heal the planet, right? So that's my job. And I've got this very sick patient that's here with us, right? And this is the Earth that we live on. If you just look at the news, you'll see tons of evidence that we live in a world that's in a state of decline, right? There's the antibiotic resistance crisis, there's the increase in superbugs, there's weird climate, including snow about a week ago, and lots of heat right now. And all of these changes, I can make an argument to you that microbes are at the core of some of them, whether it's through mediating volatile compounds, as a colleague of mine studied. You can trace microbial communities in all of these events. So this is my sick patient, right? And microbiome scientists, myself, and a few of these guys sitting here, are the healers of this planet. This is because we live in a microbial planet. There are more microbes on this planet than any other biological entity on this planet. These microbes drive ecosystem services. Whether you look at the oceans, where microbes are important for carbon dioxide recycling and taking up anthropogenic CO2, whether you look at soil, where they recycle organic carbon, there isn't any system on Earth that is not touched by microbial communities, and there isn't any ecosystem where microbial communities do not play a role. So the microbiome is quite important, and it underpins any efforts to look at One Health. So if you look at anything that happens biologically on this planet, I can show you evidence that microbes are at the heart of all of these ecosystem processes. You look at animal health, you look at human health, there is a role that microbial communities play. Microbial communities have even been shown to play a role in regulating mental health. So there isn't any kind of process that happens on this earth that doesn't depend on understanding the role of microbial communities. So our role as healers is even more important. So I like this tree. This is the phylogenetic tree of life. What this tree shows, it shows all of the biological entities that are present on the planet. You and I'm not going to touch anything, are at the bottom in green, but the vast majority of everything that's out there is microbial. A lot of these communities, especially in the clades that are on top, are microbes that are present on a sequence level. We haven't isolated these organisms in the lab. But even with these organisms, I guarantee you there are trillions more microbial communities that are yet to be discovered and play important roles. Of course, we know the role of microbial communities when it comes to human health, right? You know when microbes make you sick. The listeriosis outbreak is one such example where you know there are microbes that are pathogens, but a great majority of the microbes that are out there actually give us very beneficial ecosystem services. They're important in cycling biogeochemical elements, and they also have impacts in regulating climate change. So in this plot, it shows the United Nations Sustainable Development Goals. And when you look at all of these goals, whether you look at good health and well-being, there's evidence of microbes playing a role in terms of mental health. Whether you look at zero hunger, microbial communities are important in terms of making sure that your body breaks down compounds that it cannot digest itself. Whether you look at climate action, life on water, there isn't a single SDG that can be achieved without recognizing and studying the role of microbial communities. Right? I was supposed to transition, but yeah, there it goes. So there have been lots of advances in the past 3 decades that have allowed us to understand the role of these microbial communities. As I've said, when I showed you that phylogenetic tree, a lot of these microbes we know at a sequence level, but we've not been able to characterize them by growing them in the lab, right? There's been advances in genomic sequencing that have, especially since the advent of the Human Genome Project, allowed us to more carefully understand and study these microbial communities without necessarily having to grow them in the lab. So that's what we do. We look at microbes from a variety of different ecosystems, whether you look at microbes associated with humans, you look at microbes associated with animals, we study all these microbial communities across all of these different zones. There are lots of challenges, of course, that come with studying these microbial communities. The first is that these microbial communities are quite numerically abundant. I mentioned earlier that there's essentially, if you take a swap of this desk, right, not assuming that Alpha was here before, did something bad, but there will be likely trillions of microbes on this surface. The complication is that even when you take the swap and extract the DNA, what you see might change in a very short period. There's continual horizontal gene transfer, gene loss, and convergent evolution that makes it very complicated for us to study how microbial communities vary in space and time. We also know that all of these environments that we look at, microbial communities change depending on the environmental condition, right? So if you look at soil microbes in desert systems, the harsh climatic conditions select for specific types of microbes that might be different from microbes that grow in the back of your garden, for instance. So all of these physical chemical variables make it very difficult for us to get first a true sense of the extent of microbial diversity, but also how it changes. So if we don't know yet what microbes are there and how they change, it makes it difficult for us to harness the beneficial processes that we can derive from these communities. So microbial communities in polar and also marine environments are really good, excellent models for us to understand how microbial communities vary, but also to understand the impact of climatic change. Why is this? This is because when you look at a harsh desert, for example, the cold temperatures in the Miles Dry Valley of Antarctica limit the type of microbial communities that might grow. It limits diversity, reduces those numbers of trillions of microbial communities to a more manageable size that allows us to get a true sense of what the diversity is of these microbial communities, and also get a better sense of how they interact with each other, but also evolve over time. And once we can get a sense of these mechanisms in these environments, we can make extrapolations to other systems that we might study. And I'll give you an example of this as we go through. So I've been quite interested over my career in studying polar and marine ecosystems for a number of different reasons. The first is that they're quite cool environments, right? That was a joke, but anyway. And then the second is that we have a very strong geographic advantage as South Africa. Most of you will know we've got the Southern Ocean directly below us. We're surrounded by 2 oceans, and we have a base in Antarctica that allows us to undertake regular logistical cruises. And these logistical cruises allow us to study what happens in the Southern Ocean, which is, and now you'll hear more about it, one of the most impactful marine ecosystems globally. There are lots of strong scientific justifications in terms of our work in the Southern Ocean, for example. This environment is limited in trace elements, more limited than other ecosystems. And we want to understand how, for example, a limitation in iron impacts the biology of these systems. There's also been quite strong government support for fundamental research in Antarctic system. South Africa has been a signatory state to the Scientific Committee of Antarctic Research since its an exception in 1958. And that comes with quite strong support for undertaking research in the Southern Ocean. And we've been involved in several efforts towards establishing global microbiome and biotechnology initiatives, and we've taken quite a strong road. And this support has translated into lots of funding possibilities, including a European Union program, which has supported a lot of our research in the past few years. So I love Antarctica because it's a cool environment, and this is a system where I've had a lot of research in the past few years. And of course, in research, you don't do the research alone, right? As you will see as I go through these slides, you always see faces of people that I've collaborated with in the past that have been instrumental in driving the science that we do. Because that's the nice thing about research. We work in teams, and these teams, we make people that become friends and lifelong friends. And that's important because you can't generate and share new knowledge if you're in an environment of people that you kind of don't like, right? So it's quite important. So I'll talk you through a few studies, right? The first study that I'll talk about is work that we did in the McMurdo Dry Valleys, where we try to understand in a system like Antarctica, where there is very limited impacts and human impacts, whether we see evidence of antibiotic resistance genes. So we collected samples along a very long longitudinal transect, and then we tried to probe using metagenomic analysis what the diversity of microbial communities are in these systems using genome-resolved metagenomics to reconstruct the microbial communities, and also looked at the type of antibiotics that are recovered from these soils. What we found is quite surprising evidence that even in such an environment, you see evidence of a high diversity of antibiotic resistance genes that are present within these systems, which is really besides what you would expect, especially when you go in a system that hasn't been studied, like these Antarctic systems. So in this work, we provided some of the first evidence showing that in environments that are pristine, you see a high proportion of antibiotic resistance genes that are present in these soils. We then did quite a lot of studies, but one of the studies that we did, we wanted to understand how bacteriophages, so viruses that infect bacteria, how these bacteriophages, what role they play in terms of directing the diversity and evolution of microbial communities in Antarctica. So without going into the detail, what we show as well in this paper, which was led by Oliver, who's sitting right there, is that we see evidence of a high diversity of novel innate immune system genes that are present in these systems that are quite ancient and not impacted by microbial communities. What you do see evidence of is that bacteriophages seem to play a very profound role in structuring the diversity of microbial communities in Antarctic ecosystems. So of course, I've studied a lot, and there's been a lot of papers, so I won't go through them in terms of the work that we do in Antarctic ecosystems. We currently have ongoing projects where we're trying to simulate the effect of climate change and understand what impact, for instance, increasing temperatures or increasing ice melting or glacial melting would have on functional processes in Antarctic ecosystems. And we published a number of papers, including one published quite recently by Girish, where we showed, for instance, that microbial communities are likely to have more increased interactions due to the increase in carbon that's available, that comes about through the interactions, through the result of increased temperatures within Antarctic ecosystems. So I've mentioned that we've also done a lot of research in marine ecosystems, including a lot of different oceans that are around us. So I'll talk you through some of the examples of the work that we do. So there are really no logical scientific justifications. I'm sure there are. I can make them up. I make them up all the time in grants. But the really important one is that this is what it looks like, right? So if you put me on a boat and I have a potential to spend time on a cruise, there is possibly no other better system, right? And the fact that I can do science on a boat, even if it's 2 months and I miss my daughter's birthday, sorry, Millie, is still, much something that I would do easily again. Sorry again, Millie. So this is the system that we study. We study the Southern Ocean. This system is an important climate because it surrounds Antarctica and it also lacks continental barriers. It's the strongest oceanic current. It has the Antarctic circumpolar current. The Antarctic circumpolar current is vital for marine and global ecosystems. It's important for transporting nutrients across all of the world's oceans. Essentially, this ecosystem is the basal stage of all major food webs in the global ecosystem. So the Southern Ocean is also important because it takes up over 40% of CO2 that we produce from all our actions, and it is responsible for approximately 75% of total ocean heat uptake. So what we've done in the past 10 years or so, we've set up a program where we wanted to understand what role microbial communities play in the Southern Ocean, where there wasn't a program before we started. And what you see in that video is essentially the team that is down in Antarctica and the processes they go through in order to get water from very deep depths of the Southern Ocean, bring it up to the lab where we study it using the genomic sequencing tools that I talked about earlier. So we've got lots of different research questions that we've studied in Antarctica, but the key questions center around understanding what is the diversity and functional gene repertoire of marine microbiomes. We're quite interested in understanding how trace elements, including what I've mentioned previously, that in the Southern Ocean, iron, a very important micronutrient that is essential in terms of protein synthesis, is quite limited in the Southern Ocean. So what we try and understand is how that limitation in iron impacts on the ability of microbial communities to carry out ecosystem services. And we also try and understand how does trace element limitation influence functional gene diversity and biogeochemical cycling in this environment. So as I've said, before we started our work in the Southern Ocean, there were very few studies that have focused on the Southern Ocean. In this plot that's shown here, the top bar shows all of the work that's focused on macrofauna, so seals and the charismatic macrofauna, and the bottom shows the work that's focused on microbial communities. What you will see is that although there's been a steady increase in our studies, compared to the studies on macrofauna, comparatively little is known regarding the role of microbial communities. And given my central argument that microbes drive all ecosystem services, we need to urgently close this knowledge deficit related to our understanding of microbial communities. So we've done some work, and this very colorful tree, a few of these will come up. This is another depiction of the tree of life, similar to the one I showed you earlier. This tree of life is specific to samples that we collected from the Southern Ocean. And what you will see is in red, it shows an abundance of gamma proteobacterial lineages that are present within this environment, and this diversity is very different from other marine ecosystems. And part of our work is to understand what role are these microbial communities playing in this environment, and how might climate change impact their evolution and the ecosystem services which they provide. So the first question that I'll touch quite briefly on, we looked at diversity and functional gene analysis in the Southern Ocean. This work, which was done by a former student, Sandor Pormer, was published, and this was the first synthesis or study that looked at Southern Ocean microbial communities. So I'm not going to explain all elements of this figure, but what we tried to do is we went on this cruise, as you can see in figure A, where we collected several samples at different zones in the Southern Ocean. We looked at microbial communities across different depths, so collecting samples as deep as 5 kilometers. And what we tried to model is how do the functional processes that are mediated by microbial communities change over that distance, and how, for example, when you look at the middle, which is this layer of the ocean where you'll find fish and all the other macrofauna, how does the capacity of microbial communities to fix carbon, nitrogen, and phosphorus change in those environments? And how does the influx or the movement of nutrients along the water column influence biogeochemical cycling within these systems? So in this study, we use genome-resolved metagenomics, as Alf mentioned. Our study, unlike classical microbiologists, we don't necessarily grow most of the organisms that we study, but we look at them at a genomic level. So using these tools, we're able to reconstruct the majority of organisms that would be almost impossible to grow in a lab, right? Because they need particular biotic pressures. They need, for example, very minute nutrients that we cannot produce artificially in the lab. So using metagenomics, we were able to reconstruct these communities. And we showed for the first time that these microbial communities vary quite substantially in terms of the diversity. They are dominated by different bacterial communities and also a fair proportion of archaeal communities. So again, that colorful tree that you see on the right, it essentially tries to map what ecosystem services these communities provide. And you can see that a lot of the processes that, well, you can't see because it's quite small font, but a lot of the functional processes that we see from these communities, including carbon assimilation, are dominated by those gamma proteobacteria that I showed you were quite a dominant proportion of these microbial communities in the last slide. And we see a lot of evidence of microbes that are able to fix inorganic carbon using a multitude of different pathways. And this was quite interesting within the context of the Southern Ocean, where microbial communities drive a lot of ecosystem services. The second study that I'll talk about, aims to understand how trace element availability influences microbial community and also the evolution of these communities in different systems. So in this study, I've talked about how when you look at the ocean, right, it's essentially a very complicated four-dimensional space, right? You've got different water masses that continually circulate within the system. So what we tried to do in this study is use this instrument that's shown here, it's called a marine snow catcher, to try and simulate how carbon drawdown occurs in the water column. And we're able to suction off different proportions of these microbial communities as they settle, and specifically try to measure how microbial communities vary as they go down. And what we tried to do in the study, we showed that bacteria and archaea are quite important regulators of organic matter export in suspended and sinking particles in the Southern Ocean. So you might ask yourself, what is the importance of this study, right? So we know, for example, I've given you evidence that the Southern Ocean is quite important for taking up a lot of the carbon dioxide that we produce. What we try to do in this study is simulate how, as the climate changes, this capacity to sequester the CO2 is likely to change, and which microbes might play a more enhanced role in carrying out these processes. So this is the last study I'll share in our marine environments. But over time, and especially with my recent move to Stellenbosch University, I've become much more interested in studies that are focused on understanding microbial diversity in Africa and studying how we can translate the fundamental research from our outputs in previous studies within this context. Right, and I'll start with this slightly goofy video, and maybe give you a little narration. So when Oliver and I came back, I had done a sabbatical in the US, and Oliver had done a postdoc. We came back and we were quite interested in seeing how we can translate the work that we've done into looking at questions that are quite important within the South African context. So in this study, we went to a village in Limpopo, my name is spelled incorrectly, and tried to, in this village, they have two different forms of water, and these waters have different forms of pollution, right? What we wanted to understand, and I'll show you in this image here, so we collected waters along different transects, and we wanted to see whether comparing the traditional measures of measuring contamination in water. So typically we do total fecal coliform test. Those are the known standards of measuring quality in water. But we wanted to use this molecular marker, crassphage gene, which we've seen as a much more sensitive indicator of contamination in these waters. So what we did, we measured how crassphage diversity varies across different points impacted by different levels of contamination. And what we're able to show in this study is that the crassphage sequence is a much more sensitive indicator of contamination within contaminated waters that we studied, right? So what we want to do, looking further in this study, is to try and think about how we can essentially apply this crassphage marker into detection of water quality across the country and have this looked at as a much more useful and much more sensitive indicator of contamination of water quality across South African waters. Another study that we did, this study is slightly before the one I mentioned, is a study where we tried to understand how healthy individuals in urban and rural parts of South Africa, how their microbiomes differ, specifically how their gut microbiomes differ. There's been a lot of studies that have essentially looked at the gut microbiota of urban and rural people in the global north, but very few studies have focused on people from the African perspective. And what we did in this study, and when they have looked at African individuals, the studies have tended to compare, for example, African American individuals to the Hadza population in Tanzania. And we feel, of course, knowing that genetic variations and a range of other factors influence the gut microbiota, these are not really fair comparisons. So in the study, what we did is that we sampled stool samples. It was not very easy, from people in the Vembe district of Limpopo and also in Pretoria. And we made sure that from these individuals, we looked at the variation of bacterial communities, but also of fungal communities. I'm not going to talk about the study that focused on the fungal communities, which was published previously. But what you will see from this study, and I'll share in the next slide, is that although you see more or less similar proportions of microbial communities in urban and rural South Africans, when you look at these complicated network analysis plots that are shown, is that if you look at the rural samples, you see that there is a lot more connectivity in the nodes. So essentially, in each of these plots, each dot indicates different microbial communities. So the connectivity essentially shows you that the microbial communities in the rural communities and the urban communities, it tries to mimic how these microbial communities cooperate, right? And what you will see is that people from rural locations seem to have a lot more connected nodes compared to individuals in urban populations, right? And what does this imply? When you look at the urban microbiota, there's a lot more disjointed connectivity. You remember I said the phylogeny is more or less similar. So what this implies typically is that a disconnected microbiota, which is characterized by less connections, indicates a state of dysbiosis. So it's an unhealthy microbiome. And we try to test what is the implication of this lack of connectivity. So we use antibiotic resistance genes as a marker or a functional proxy to determine what the effect is of these two different or disjointed connections. And what you will see is that when you look at the rural microbiota that's shown on the left, is that you see less antibiotic resistance genes compared to urban microbiota, which have a higher number of antibiotic resistance, right? So this, of course, validates the network analysis that we've shown previously, that in rural individuals whose diet is typically characterized by processed foods, a much more unhealthy diet, you see this manifest in an unhealthy microbiota within these individuals. Whereas in people that are living in rural locations have got a much more healthy microbiota. This translates to, of course, a much more stable microbial community, gut microbial community in these individuals. So I'm going to take you quite quickly through some of the lessons that I've learned from studying environmental microbes. So the first lesson that we've learned is that when you look at microbial communities in the oceans or in Antarctic systems, is that it's important that you take into account the environmental context in which you study these microbes. We know, as I've mentioned previously, that depending on the type of system that you look at, it can quite disproportionately shape the diversity of microbial communities in the system that you're looking at. We know, again, that when you look at microbial interactions are more consequential than single microbes. So in the study that I showed you, where I looked at the interaction between the gut microbiota of urban and rural individuals, so often what most people do, they look at isolating particular organisms, maybe probiotics, and infer the importance of those microbial communities. But often, it's not only the single organism that plays a role, but it's actually the interaction of that organism and the microbial communities around them that's quite important, right? It's quite hot in here. So, and then the third is that when you look at microbial diversity, high diversity leads to community stability in these systems, right? So having a microbiota that is quite high in its phylogenetic diversity is really important, because if something were to happen that disturbs the communities, there is a lot of functional redundancy in the communities that you get, and the process that might be wiped out by the removal of a certain microbe can easily be replaced by another microbial community. The fourth example is that microbial communities are subject to successional patterns, right? There is a lot of variability in microbial community, especially when you look at them in the environmental context. The fifth and very important lesson is that changes in environmental conditions influence, I'm going to take this off because it's really hot. Julia, do you want to take off my tie? It's quite unusual, but I'm getting hot. You can't take it off as well. Okay.
Comfortable like that?
Yeah, it's more comfortable. You are. So changes in environmental conditions. So I'm going to talk a little bit about what I hope is our future prospects. It really is hot, but this was part of an act. So I've talked about how our work has slowly transitioned towards looking at African microbiomes. And there are several reasons for this, right? This plot shows the demographic projections over the next 100 years. Thank you. So what you'll see is that the population on our continent is set to increase quite substantially compared to the population in other environments. So what is the implication of this? Well, we are likely to have an increase in the number of young people, and studies on the microbiota have important consequences. And I'll make this point this way. So we know that a lot of the studies that we've looked at have tended to show and focus on populations in the global north. Very few studies have actually focused on understanding variability in the microbiota of African individuals. So if you take, for example, efforts to have precision medicine, we know that genetic diversity is a strong influence on the gut microbiota, as one example. So if you wanted to, let's say, develop precision medicine tools, you cannot do this without understanding the variability of the gut microbiota of urban individuals. So with this plot, I'm trying to really underline the more critical importance of studying particularly African individuals. So it's become a very big theme within the research that we do, and a lot of the studies that we're more or less transitioning to are focusing more and more on elucidating the diversity of African individuals. So here, I'm showing a plot of a perspective article that we published last year that outlines some of the steps that we hope to take in terms of reducing the current knowledge deficit related to African microbiomes. The first is that we've called for establishing, and maybe before I show this, the plot that you see there with the yellow and green, right, essentially shows you the available sequence data that are stored on the current databases. What you will see is that on our continent, there's a huge gap in the current knowledge. There is very little work that's focused on the microbiota of African individuals. And what we are doing, or what we hope to do, is essentially propose a roadmap on how we can reduce that knowledge deficit related to studies on African microbiomes. The first is that we've worked quite strongly towards developing coordinated research networks that are continental-wide. And part of this is that when we start to form strong networks, train core researchers that are interested in understanding African microbiota, working together in teams, we can work towards reducing the knowledge deficit on African microbiomes. The second is that we're more or less working towards writing or developing specific policy interventions promoting the increase in research focused on African microbiomes. Whether you look at, for example, innovation policy, or how samples can be retrieved from different parts of the continent, there is a lack of a policy framework directing how this research should be done. And the problem with this is that it's become an opportunity for others, typically from other locations, to study African individuals and African samples without necessarily respecting the local communities or incorporating them in their research. What we hope to do in our group, working with others, is to more or less start developing the policy framework that would underpin the work we want to have on African microbiomes. And the third, this is also another important reason why I've moved to Stellenbosch University, is that we are calling for the coordination of specific infrastructure hubs across the continent. And we're trying to work with SERI and others in order to develop some of these research hubs that would promote the sequencing of African microbiomes at continental scale. So I've given at the bottom as a perspective, concluding remarks by Rita Colwell, who is one of the leading microbiome scientists, at a meeting that happened in 1993. And in this meeting, she noted that the perspective titled Biodiversity Amongst Microorganisms and Its Relevance, she makes the point that we are at a fortuitous conjunction of events with developments in biotechnology advancing rapidly. The tools and techniques of microbial ecology have reached a maturity which allows research to be conducted which could not have been envisaged even a few years ago. It's quite ironic that this statement was made in 1993, but you could make exactly the same statement now. But we make the statement within the context of the African context and in terms of enhancing the work that we do on African microbiomes. So in our group, I'll just give you a brief overview of the research that we want to transition to for the next few years. We have essentially 5 thematic areas. The first is a theme which focuses on the research that we hope to do. And you'll see in this theme, which is focused on discovery and innovation, we want to have an increase in the number of projects that are focused on the dynamics of African microbiomes. Understanding climate change effects on African microbiomes, looking at African environmental surveillance. We've got a project that JP has envisaged where we're looking at wastewater systems at over 12 different African countries. And we hope to set this up as a long ecological monitoring project, looking at antimicrobial resistance across the African continent. And we've got a strong research theme focused on microbiomes and health, of which I don't have time to talk about today. But one of the core things that we want to do within the group and within the broader context is focus quite strongly on training the next generation of African microbiome scientists. And we've, Alf says I'm on a plane every week, and part of the effort is to build that network of people who will work with us in order to make sure that we have the resources that are required towards training the next generation of African microbiome scientists. We focus quite strongly as well in terms of developing microbiome services and also support. And we're also working as part of our efforts within the group to launch spin-off companies that are focused on the fundamental research that we produce within the group. We're also conducting, as I've mentioned previously, coordinating microbiome policy and education throughout the continent, and we're working as well towards strengthening regional and international networks focused on African microbiomes. So this is the work that I hope will occupy my efforts for the next 20 years, and hopefully you're here to witness as this research groups. So there's sort of 5 major areas that these studies will cover. The first is we're quite interested in understanding the extent to which evolutionary factors such as gene duplication drive genome expansion and what the implication is on microbial communities. I talked about how microbial community interactions are important. The second core theme is that we're interested in understanding how interactions within microbiomes and what is the influence of these on biogeochemical cycling within a broader range of ecological context. We're working quite strongly towards efforts in terms of measuring and integrating community-level data into process kinetics, and also using these data to develop herb system models, for instance, in the work that we do within the Southern Ocean. I've talked about how we look at phylogenetic diversity, but when you look at a lot of the models that exist, looking at CO2 fixation, for example, none of these models integrate microbial communities. And part of our work is to try and incorporate these into these Earth system models. We're also working on using artificial intelligence tools to conduct large-scale analysis of function within microbiomes. We've got a program where we're looking at biosynthetic gene clusters and trying to think about how we can commercialize some of the fundamental research by using or applying some of these tools. And lastly, we are looking at understanding how viral host interaction impact ecosystem function across a variety of different functions. And this really broadly are the 5 core major areas that our future studies will touch on. So I talked about how we as microbial ecologists are the healers, right? So towards the end of the talk, I'll end off by giving what I think is a prescription for our sick planet. The first is that scientific questions that drive the research questions that we do, they can never supersede the obligations that we have as global citizens. The second is that the work towards improved microbiome literacy, expanding the current network of researchers that are focused on understanding microbial ecology is important. So if we can grow the pie of skilled individuals that can study microbiomes, it's quite important and fundamental towards reducing the knowledge deficit on African microbiomes. And the third is that we must work towards improved access of genomic capabilities, which will expand the current network of healers, of microbial ecologists that are undertaking the research studies that I talk about. So with that, I'll bring my presentation to a close. And I think my grandmother is half awake, which is possibly good. But with that, I really want to take some time to make a few acknowledgements. I'll start with the Stellenbosch University Rectorate, who have really allowed our group to thrive since arriving here. I think we've received a lot of support, and I am really thankful, and I look forward to continued support that will allow our group to thrive. I'd like to thank my dean and the faculty that have also supported us, and it's been great having you here. My dean takes a lot of shots at me. I don't know if I should contact HR, but I will think about it. And then I also want to have a special acknowledgement to my colleagues in the Department of Microbiology, who have really also welcomed us quite openly since our arrival. We've received tremendous support, and I'm really thankful for all your efforts. I'd like to thank Kanshu and the School for Data Science as well for being a fantastic pillar. Can I call you a pillar to lean on? And I'll thank as well Tulio and Frank as well from the center. It's been really nice to come into an environment which is research-driven and quite passionate, particularly about driving the research that we do throughout the African continent. And I think it's been a very good combination, and it's been very good support for our group. And I hope people from the University of Pretoria are also online, because although Stellenbosch stole me, I think a lot of the work that I presented, would not have been possible without the support of the University of Pretoria and my colleagues in the Faculty of Science, and I'm really grateful for all the work that they've done. And also, I extend the same gratitude to my colleagues in the department that I was linked to at the University of Pretoria. I won't link all the national and international collaborators by name, but as I said, the work that we do really depends on the teams that support the efforts that we do, and none of this work would be possible without their support. These are just some of the former colleagues and friends that I'll never forget that have supported all the work that we've done. In coming in as a new academic, having support in terms of the research management, in terms of supporting grant applications, but also having scientific support has meant quite a lot to me. I mean, I have Colin here, who's been a very good mentor and friend over the last 10 years, sadly, and I'm really grateful for the support that he has given over these years. Again, I really want to thank the former students who have contributed to the research that our group has produced. None of the work that I talked about would be possible without their efforts, right? And then these guys as well that surround me, I can't really thank them for all their contributions yet, but I can anticipate that they will contribute quite substantially to our efforts in the future. I really hope so. At the moment, when I look at them, all I see is this. Especially the honor students, that cost us a lot of money. I've removed $1 for Sam, who just got his first grant for R1 million today. So that's the kind of example that we want. And I'll thank my family. As I said at the start, I would not be here without my mother and father, my grandmother and extended family, who recognized that I had a little bit of potential and nurtured that potential. I had all the tools that I needed to study. I never really needed any academic tools because my father would make sure those resources are there. And I think I am here because of their efforts and their sacrifice. You can see, especially if you look at the second picture, when my dad graduated, he really modeled being a lifelong student. I think this was when he got his BA. You can see from the flip-flops that times were a little bit tough, but we made sure that we put education at the center of everything that we do as a family. And I hope to model that as well to my kids that are sitting there. No pressure. And I want to end off by thanking the love of my life, Andy. So, the whole line that behind every man is, what do they say? You said it, can't you? I really would not be here without an intelligent and super beautiful wife. So, and I'm really glad for all the support that she gives me and my family and our kids. We really would not be here without her. I've always been able to depend on her on everything. If I say, maybe we should try move to Stellenbosch, she just says, if you want to move to Stellenbosch, let's go. And she's there to support all my efforts. And my two girls, Papa loves you. And I'm super proud of you. You know that, right? Thank you for staying awake. And I look forward to seeing you guys as well one day give your inaugural lectures in the stage. With that, I'll bring my presentation to a close. And thank all of you for being here and for sitting through this lecture. but really thank you more than anything for the support that you've given me and my group since we arrived at Stellenbosch University. It's been a brilliant 10 months, and I look forward to what I hope is a bright future at Stellenbosch University. Thank you.
Thank you so much, Thulani, for that excellent lecture that you've given. So my name is Sibusiso Moyo, the DVC for Research, and of course, it gives me great pleasure to make just a few remarks about the lecture and then hand over the certificate as well. I want to observe all protocols, and especially also I know the incoming Rector and Vice Chancellor, Professor Dinesh Ramjugunath. Thank you so much for joining us. And of course, Thulani's family, the colleagues from the faculty, and also the dean, the head of school, and the students especially who are here, and colleagues who've traveled also from afar to join the lecture. You would agree that it was an excellent lecture, right? Yes? So now, now we know the importance of the microbiome. I feel sorry that our rector, the current rector, is not here because he really missed a chance to use his gut jokes as well, and the importance of the microbiome. So of course the lecture we know emphasized the incredible diversity of microbiomes in various environments, including of course the human body, animals, and how these microorganisms influence health and disease as well. And then the issue on interconnectedness, the lecture emphasized the fact that microbes underpin the One Health concept, yeah, which also talks about all the importance, the important connection with the ecosystem services that we see. He highlighted the interconnectedness between the environment, microbiomes, human health, suggesting that changes in the environment can significantly impact our microbial communities as well. I was a bit jealous to see the Antarctica pictures, but I see that the ice is melting, right? So it would be interesting to understand the impact of course, climate change on also microbiomes, which I think you've also touched in your talk. So the importance of studying microbiomes for applications in medicine, agriculture and ecology, you know, and other areas is important, and we know that these studies, of course, have benefits for understanding measures towards prevention and also improving our environmental sustainability. There was a close link to the Sustainable Development Goals. And also you did talk about, I think, the potential for personalized medicine based on an individual's microbiome composition, and as well as, of course, future directions that the research intends to take. So for me, I think the main takeaways are really trying to, you know, the way you've mapped out the future in terms of understanding the broader implications of microbiome research on our understanding of the One Health concept. And then also, you know, the type of work that you want to do in terms of really building and understanding the African microbiome, which I think is important in terms of setting the agenda around the African microbiome, and the potential also for spin-offs. And I know when I visited ALF, I've been here for 2 years, a bit longer than Thulani, but I remember visiting ALF. He's got this, ALF is Professor Alf Botha is the head of department for the department that Thulani is in. And he's got this special presentation where he shows off the spin-offs, right? So we are really excited with the potential as well. I could see, like from your presentation, the potential for translation, using you know, the research that you are doing to also make sure that we have some spin-offs and potentially the students who can take part in that as well. I think you forgot to mention in your group as well, your last photo, the $sign. I think you've just got a laureate, women laureate fellow winner. I don't know if she's here with us from your group. I'm sure that will come with some dollars, so you have $2 off. At least that will help. Yeah, but without saying much, I think the lecture essentially, you know, points out the lecture has given us a visual, not just visual link of microbiomes to essentially, and how these are linked to the various SDGs that are being addressed, making the point and understanding the ecology and the evolution of these communities is key to achieving some of the Sustainable Development Goals. And The next 5 years, of course, will focus strongly on SDGs 2 and 3, with studies focused on improved nutrition and good health and well-being. You might have not exactly said that, but at least it's implied in the talk as well. So we want to congratulate you, Professor Thulani Makhalanyane, for delivering the 2024 inaugural lecture once again at Stellenbosch. I know you did this at University of Pretoria. I hope that this will be your last one because you promised not to move. So we want to thank you for your lecture, and we wish you the best in the next 5 years. May I then present you with a certificate, and may the audience clap, please, at this point. Thank you. Thank you.
Ladies and gentlemen, my name is Kanchkan Rajaratnam. I'm the director of the School for Data Science and Computational Thinking here at Stellenbosch University. First of all, congratulations, Thulani. Do I have to call you professor now? So congratulations, Prof, from Faculty of Science, from the department, of course, the School for Data Science and Computational Thinking. I see the leadership from both SERI and the School for Data Science have been here to support this, so we are very proud of you. We saw his research and we saw what the kind of publications and the impact he's had. And without a doubt, that is amazing. But what is also amazing is how in his presentation, he's shown light on his collaborators and his former students and students. And I think that speaks of big volumes about you, about how you collaborate and how you uplift others to the point where you are. So, I think you should take great pride in that. It also you're standing between me and drinks, so I won't take long. Usually when I hear a science lecture, I want to have a lot of wine. But here, I understood what he was speaking about, so I want to have champagne to celebrate me, right? Pardon? And wine, but champagne more. I want to thank all of you for coming here, his family especially for coming here. Thank you to the DVCs who are here, the dean, the head of department, as well as the vice deans from science, his colleagues at the school and SERI, as well as all the students and his collaborators here. There are lots of people who made this possible, so let me take a moment to thank them. Suzette Els, who works in the dean's office, I think I saw her earlier. Marietta van der Warm, who is the director of the Faculty Management in Science, Amira Brown, Chuma Dias, I've got very bad eyesight, Dialaka, Justin Alberts from the communication team, and of course Jonathan Dale Blackenberg, who keeps us narrow and straight and makes sure that we do our job and who helps to organize this. So thank you, Jonathan. We invite you all to come and have drinks, and there's some eats outside. Come and celebrate us, especially with champagne. Can I ask everybody to stand up while the procession leaves first, and then you may join us for drinks. Thank you, everyone. Congratulations again, Professor.